to the Weekly Bioanalysis, a KCAS podcast.
Hello and welcome to the 85th episode of the Weekly Bioanalysis, the official podcast of KCAS Bio. KCAS Bio is a bioanalytical CRO serving the pharmaceutical and biopharmaceutical industries for over 40 years. My name is Dominic Moreno, and I've been in this field for close to 30 years. I'm here with my co-host, John Perkins.
Hi, I'm John Perkins, and I've been in the bioanalytical LC-MS space for around 30 years.
John and I are members of the growing scientific advisory team here at KCAS Bio. Either or both of us are available to answer any of your questions you may have regarding this podcast or any of KCAS Bio services. Today, we are here with a very special guest, Neil Spooner. Neil, please introduce yourself.
Hi, and thank you for having me today. My name is Neil Spooner. I'm the chair of the Patient-centric Sampling Interest Group. I have a number of other hats as well. I run a consultancy company, and I have a visiting position at the University of Hertfordshire as well. And I'm the editor-in-chief of Bioanalysis Journal.
Neil, thanks for that intro. We're excited to have you here. We have a jam-packed show. So we are ecstatic to have you listening to the 85th episode of the podcast, which is now available virtually everywhere. Wherever you choose to find and play your podcast, you can now likely find the Weekly Bioanalysis. We welcome all of you, whether you are joining us for the first time or if you're a regular listener. Today's podcast will be a review of the latest news and resources, and then a focus on a topic of our choosing before discussing any feedback we've had from you. We're constantly looking for topics, and we'd be happy to discuss something that you want us to cover. So again, we're fired up to have you here, and we are looking forward to a fun episode. To kick us off on podcast 85, John is going to go over today's podcast topic a bit before we jump into the news and resources. John?
Hi, as usual, we'll talk through a few medical research news items, and then our main item with Neil is to talk through patient-centric sampling, expanding sampling beyond the clinic and the potential logistical and bioanalytical challenges. Let's move to the news.
All right, John, why don't we just hop right into the news and resources? Why don't you kick us off with the first item today regarding Roche?
I'm going to say ahead of time. It's like everything is a little bit in sort of slightly torporous mode after the holiday. So the news section has been quite light. There's talk of IPOs and stuff coming, but nothing, not too
lots of that though.
Interesting stuff. I mean, it's good to watch, but that's it. Anyway, this is harking back a little bit to my past. In that when I worked for Quintiles, or when we were when I worked for Advian, who were bought by Quintiles, I was lucky enough to go and visit a couple of Quintiles facilities, which are, doing the clinical, all the clinical testing to support clinical trials. And you walk through the lab, and it's just all these masses of huge instruments, a lot of them designed by Roche Diagnostics. But the one big gap was that the mass spec on their sites was virtually non-existent, apart from doing the odd tests like testosterone or something like that. And this article is basically Roche Diagnostics, that is, are very focused on having a suite of instruments to do all testing. And this is Roche to bring mass spec to routine labs with new cobas systems European approval. Now, the article is very light on detail, but it's basically they've rolled out a new diagnostic system that aims to bring the gold standard accuracy of mass spectrometry to the everyday clinical lab. So it's a cobas i601 automated analyzer, and it has 4 assays that they're currently set up to run for tracking steroid hormone levels is in the first steps of a planned global launch. So it's not available in the US yet. They want to eventually go to 60 analytes, including vitamin D metabolites and immunosuppressants and other therapeutic medications. It's like I said, there's very little detail because according to Roche, the system required the development of a new proprietary chemistry to make an automated mass spectrometry workflow viable for commercial use. Previously, the approach was the domain of specialist labs due to its hands-on complexity and longer turnaround times. So I can only assume they're taking LC out of the equation. I'd love to see what's behind it. There was actually, when I first saw this announced, there was a really slick video talking about how they're fitting this cobas thing into the flow of the whole cobas lab environment. And for me, it's interesting as a mass spec person to see like I say, harking back to 2011, 2012, where there was no mass spec in the big labs, apart from in a little side room doing the odd test. Now here it is, as Roche at least have found a way to start moving in that direction.
Yeah, it's great stuff, John. Right. So, Roche has got plenty of immunoanalyzers, right? Those are,
Yeah, absolutely.
yeah. And so typically most of these tests are done by, I think of them as chemistry analyzers, immunoanalyzers. Beckman makes one, right? Roche obviously does. Siemens has a bunch.
Sure.
Know a little bit about this from my days at Streck Labs. We used to make a lot of controls for these things there. And even going back to the 90s, worked on Roche had an apheresis machine that was called a Cobas too. So got some familiarity with that where I don't know how much you know about apheresis, but it's like, that's what they use for CAR T cells. They draw a lot of your blood, pump back in the red blood cells and some of the neutrocytes. They try neutrophil, neutrophils and granulocytes and everything. They try to just harvest your white blood cells. And so, what I'm just rambling on a little bit, but this is fascinating, John, in that I feel like a lot of it is, if you are familiar, I've mentioned it before, like the immunocap, right? Are you familiar with that one at all?
Nope.
And that's what they use for testing like IgEs, measuring immunoglobulin E, or even like IgG specific for peanut, IgG specific for all sorts of allergens and stuff like that. And those are big immunoanalyzers. So, and that technology uses like this proprietary sponge in there so that you can get tons of capture antibody onto the sponge. And then you take advantage of the immunoanalyzer, the chemistry analyzer, for the sensitivity. So I'm not saying that's what they do here, but I imagine there's some sort of, as you said, proprietary thing that's allowing them to kind of compact the mass spec. You're the mass spec guy, but it's hard to imagine. Sorry, it's hard to imagine they can get all the make it nano size, right? That I don't think we're there yet, right? So maybe you can comment on that.
It's certainly not. If you look at the illustrations, it's a big beast. So it'll be, again, it's sort of fairly typical. And I know Neil has something to say.
Yeah, I think it's really interesting. It's going to be interesting to find out what the mass spec is that's buried inside there.
Yeah.
Because I don't imagine they've developed their own, so they've got to be using someone else's. So it'd be interesting to find that out. And it's also just interesting, I think with what we're going to be talking about later with patient-centric sampling and microsampling, this offers an opportunity to rejig workflows for these samples from patient-centric sampling and microsampling that are in a different format and are in smaller volumes, etc., rejigging those workflows, because that's one of the complexities with patient-centric sampling at the moment for diagnostics, trying to fit it into the existing workflows that work with standard blood tubes. We've got an opportunity with this kind of new platform to redo the workflows for alternative sample types.
Yeah, that's a great point. I'm happy to talk. I think we're going to talk more. You brought up a very good point, but go on, John. Yeah.
One of the other things I was thinking about, I mean, we know that obviously with repeated use, your mass specs need cleaning, etc. And again, it's if I don't know if there's derivatization going on here. What is being introduced? What size of sample? What's the constituents, and what's the cleaning protocol for this system? Would be very interesting too, because anyone running mass spec on a day-to-day basis, that is an important part of your whole daily routine. So yeah, like I say, I thought it was certainly worth highlighting, because it's a step into a different direction for mass spec. It'll be interesting to see where it goes, and it also starts me thinking again. I remember back in the day when I was at Advian, that time the lab was building. We were both a CRO and a lab building a nano source for the front end for the mass spectrometer, and ultimately when we broke out, that group went off to build a mini mass spec, because their sort of ambition was to have a mass spec that could be in a doctor's office. You'd be testing at a doctor's office. So this may be an approach that actually maybe it's a halfway step to where you do have, to talk about Neil and miniaturization. Maybe it is that step to miniaturization, such that it does become mass spec far more public access.
Seems like it, right? This is a good step. Go ahead. Sorry.
Yeah, so it could be really interesting to see what direction this all goes in. So
Yeah, it's really fascinating, John. Maybe we can move on just for the sake of time, but
Yeah, absolutely.
that evolution of the thought of being able to, because I've toured a few different like reference labs, university labs, they got almost like literally half a mile worth of instrumentation. A sample goes on there. You punch in what the doctor ordered, and then it goes across like, a conveyor belt, and then whatever instrument, mass specs being part of that are not routine now, right?
Absolutely,
This
yeah,
is the
yeah,
first step towards that.
yeah,
And then,
yeah.
like you said, the thought of miniaturizing it is fascinating. And, we talk about this often. It's great to be in science, right? I believe in our lifetime that we can make a mass spec that will be able to be more routine. And this team seems to me be the first step in that direction, although it might be the size of a Volkswagen today, right? This
Yeah,
would
yeah.
seem
Like I say, it's like I say, they're very there's very few details, so it would be
Mhm.
nice to know what's going on inside it. But it's one of those things. Ooh, I like
Yeah.
the look of that. Let's see where that goes.
It's cool though, right, John? Like that is
Yeah,
the thought of having a routine like that, it just brings global health gets amped up a little bit if you can do high sensitivity mass spec for personalized medicine, or as we'll talk today, even as we further down into like microsampling, which hadn't even thought of that. I can't wait to have our main portion to talk about that. But Neil, if you have any last thoughts before we move on to the next article here, you want to close anything out, or you have anything you want to say
No,
about
I just think it's a really interesting discussion. I'm looking forward to learning more.
it? Yeah. Well, all right, John.
let's
Why don't
move
we move
on.
right along? Yeah. What do we got next?
So, so this is Cereso claims a phase 1b success for oral antibody in ulcerative colitis. So, a private startup from Salt Lake City is aiming to develop oral antibodies. Says it succeeded in an early trial, a potentially significant scientific advancement in a world where systemic treatments are the norm. This is basically they had an oral bispecific antibody targeting TNF alpha and IL-23, and it hit safety and efficacy benchmarks in 22 patient phase 1b trial in ulcerative colitis. What's interesting, I mean, it's obviously the oral bit is the interesting piece,
Mm-hmm. Mm-hmm.
because they talk about oral antibodies have been tested, but ultimately your stomach chews them up and nothing happens. But the approach that Cereso have taken is they've basically gone for a much smaller antibody, and I think it's about a tenth the size of what you and I would consider a
Mm-hmm.
standard antibody. So make it like a 15,000 molecular weight type of thing, and so it's they're just engineered to be resistant to the proteases that bigger compounds are more resistant to, and so that's I just thought this was something to punt your way and get your thoughts.
Yeah, John. I mean, it is quite a breakthrough, as you mentioned. Most, any monoclonal bispecific, most biologics are injected. So and there are some oral versions of it, right? But most of those oral ones are monoclonal, and they all are targeting what I'll call like mucosal immune system, from your mouth to your anus type of stuff is what it covers. And in this case, that's exactly what they're attacking, right? This
Yep, yep,
bispecific
yep.
binds TNF alpha, which I don't know. I hope everybody who's listening kind of knows what that is. It's a hallmark of inflammation, right? It's something that's really anybody with like an inflammatory bowel disease, which is what Crohn's and colitis and other things are. They all have high circulating levels of that. And then IL-23 is an interesting one. It's part of the IL-2 kind of pathway there. It has some properties around, just again, more in that inflammation pathway, kind of trying to stop more. So the TNF alpha, the bispecific mopping that up just stops the action of the TNF alpha. So to try to alleviate the inflammation, mopping up IL-23 is trying to stop the adaptive immune system. So it's like a double whammy. Dangerous space too. I mean, these are when you start hitting kind of naturally occurring and I say dangerous, that might not be the right word. If you suffer from these things, you're going to want them, right? Let me rephrase that. It can just make you prone to opportunistic infections if some of your key immunological factors, key cytokines are being kind of sopped up and sequestered by these bispecifics. So John, you got like a three-headed beast here. You got oral version of it. They shrunk it by 1/10, which is fascinating, right? I'm not quite sure how they did that. And,
Indeed,
and
yeah.
then you got a bispecific, which is still, John, I bet you if we looked at the number of drugs that are approved in the biologic space, there are some bispecifics, but we're going to be dominated by monoclonal antibodies. And now most people are doing the ADC space, right? So it's just, these are really powerful drugs, not just bispecifics, but trispecifics and things like that. So just great science here, John. look, and I'll end with this. I'm a little bit hesitant to think that this is going to work as effective as like an IV administered bispecific. But hey, if you're like I said, that's why I caveat it with if you're treating the mucosal immune system, there are ways, and we've researched some of it here in some of the projects we've done, where you can alter the biologic to sort of stick to some of the mucosal cells, so they target it, which is another. And not that they get internalized by them, but they got like certain ways to just stick, so it doesn't pass right out you. You know what I
Sure.
mean? That's a problem with an oral, right? It just comes in and goes right out. It has to get absorbed. That biologics normally aren't absorbed through your gut. So that's why they shrunk it. That's probably some passive absorption there.
Yep. Yep.
But then I guess I'm kind of commingling. Others have tried ways, not shrinking it, but altering the monoclonal or the bispecific to have some sort of way of binding and kind of just stay in there. And now it's kind of on the surface of some sort of mucosal cell. So that's my thought, John. It's fascinating. Love this stuff. Really cool. I don't know. It is definitely something that it could revolutionize things a little bit for those that suffer from inflammatory bowel disease or any sort of gut kind of disorders, which are pretty high rates, John, right? Like it's amazing. I think like 30 or 40% of the population could have some sort of problem in this area, especially in an aging population, which
Sure.
is what I feel like this morning. No.
So they basically say they have a phase 2 planned for 200 patients, and they've also got other similar designs in development for other indications. So,
Yeah.
Interesting. See where this goes. Let's move on.
Sounds good.
So this is another stepping even further back into my past.
Oh.
So when I was a postdoc, this is like 89 to 92, was when electrospray was really starting to move forward. Ion spray was still really a research tool rather than a commercial application. And electrospray, one of the limitations, although it had opened up mass spec to higher molecular weight molecules, one of its limitations was it only could take a flow rate of 50 microliters a minute max. The lab I was postdocing at had a Vestec source, which basically fed into VG12250 single quad mass spec. And so we were, as in common with many labs, we were saying, we have this interface, what can we do with it? It's how do we demonstrate the utility of this source? I was at NIHS in North Carolina, and one day in the library I was going through a book, and I just picked up something at random. It was talking about snake venoms, and looking through some of the figures of that, it talked about snake venoms, particularly from elapid snakes, where the proteins were lysine, arginine, histidine heavy. They were more polypeptides, but molecular weight's around 7 to 8,000, which when you looked at the number of positive charged amino acids would bring them down into the mass range of the 12250, which was only 1200 at the time. Because what we were trying to do is actually not so much the mass spec, but we were trying to show applications of capillary electrophoresis with mass spec. So this was, how do you separate because venoms are basically a complex natural mixture of peptides and proteins. How do you separate that and then detect it? And that was what we were looking to do. So I basically ordered a bunch of venoms from like Sigma and got like black mamba venom and spit and cobra venom and lots of stuff. But they were all mostly lapid snakes, mostly with these like polypeptides in the 6,000 to 7,000 molecular weight. And so I've always been fascinated with them ever since, just from this, like what's in them, etc. And this is actually to actually now transition to the news article. Snake bites, you don't really hear about it, but there's still a massive public health threat in many developing regions. Obviously in sub-Saharan Africa, South Asia, Papua New Guinea and Latin America, with over 2 million annual cases, snake bites result in 100,000 fatalities and 300,000 permanent disabilities. In 2017, the World Health Organization listed snakebite envenoming as the highest priority neglected tropical disease. But there's been very little attention paid to this. And this is all a publication in Nature now. Mostly therapies for snakes rely on plasma-derived polyclonal antibodies from hyperimmunized animals, complemented by medical and surgical care. Although instrumental in saving lives, antivenom accessibility is hindered by high production costs, inadequate cold chain infrastructure, which will play into the microsampling piece later on, in remote areas. And then there can be serious side effects. Now, the reason why I touched on this is these were a couple of groups at University of Washington, Seattle, and Technical University of Denmark, Konings Lyngby in Denmark, as well as a number of institutions, including the Liverpool School of Tropical Medicine. And so a lot of the inefficacy of antivenoms actually for lapid snake venom, so like this is mamba, cobra, coral snake, a few others, is basically the neutralizing antibodies are suboptimal against the three-finger toxins. And I assume this is related to the fact they're all pretty low molecular weights, so they're not really causing much of an antibody reaction. And so they have like failure to elicit a strong antibody response. And these three-finger toxins are highly toxic components of elapic snake venoms that can cause diverse pathologies, including severe tissue damage, inhibition of nicotinic acetylcholine receptors, resulting in life-threatening neurotoxicity. The news piece of this is these groups have actually they've used deep learning methods to design de novo proteins to bind short chain and long chain alpha neurotoxins and cytotoxins from this three-finger toxin family. And so they've actually got protein designs with thermal stability, high binding affinity, and they actually have worked in a mouse model and actually protected mice from these lethal neurotoxins. I just thought that it all plays into my obsession from the past, but it's absolutely fascinating.
Yeah, John, no, thanks. It appreciates some of the background there, the history of it, even just snake venoms and even like poison ivy antigens, John, right? They've been back in the day, right? That's some other things. Just as like therapeutics, if I could get these types of things into like a tumor, could it cure it, right? But like back to the article here, and again, thanks for like, there are some fascinating things, John. I did not realize there were 100,000 fatalities due to snake bites.
Oh, sure.
That's unbelievable, right? And then 300,000 permanent disabilities. That's crazy to me, right? And then so you kind of touched on it. The traditional way, traditional antivenoms are hyperimmunization of animals, and they get plasma-derived polyclonal sera, which, by the way, John, right? That's what we use for an ADA assay, essentially, right? That's the positive control for an ADA assay. It's also not in mice. It's a way to treat. I mentioned primary immune deficiency earlier. Actually, I think I mentioned IBD, but primary immune deficiencies, the treatment is IVIGs, intravenous immunoglobulins. It's kind of similar to these antitoxins, only it's not obviously against like the venoms, right? So there's those types of treatments are incredibly expensive. You mentioned the cold chain effects. They're just, and it's just, they're not that effective either, are they, John? Right? So they might bind up some of these three-finger toxins, but they talk about how they have these other kind of, they call them fortifying agents, I guess, or no, sorry, they have other things within the venom that linger. So they talk about how it's not the actual toxin, and it doesn't cause any. So they're low molecular mass toxins with limited immunogenicity but high medical importance, right? So that means that there's still some toxins in there that aren't mopped up by the antivenom but cause lingering effects. They don't really know what that is. I think that's how that reads. And so these now, fast forward to like today in this paper here, John, these are, they use what I would call AI essentially, right?
Oh, absolutely. I would agree. Yeah.
And we should give a shout out to Denmark, right? That's the University of Denmark, University of Seattle. That seems to be where these are coming from. And then what do? They generated just kind of mop things that could bind to them, right? So
exactly.
Them.
Yeah.
And bingo, right? This is how you get a kind of an advancement that could really help global health. Now, is it 5-10 years away? I don't know, but I'm optimistic that this type of approach will finally, really help a lot, because it's not. I'm surprised by the number of people dying, but I'm not that surprised due to the lack of antivenom and even the efficacy of the antivenom. by the time you get it,
Sure.
sometimes it's
Yeah.
too late. So fascinating stuff, John. And you always dig up these great stories. And I think you went back to what, 89? Is that what you just said? Oh my God.
I think it was around 1991 was when I was doing
That's
this.
awesome.
I mean, it's basically, I just want to touch on it. These neurotoxin binders demonstrate comparable potency, the best in IgG antibodies, nanobodies, stable, readily producible in microbial systems, and they are small size, approximately 100 amino acids, which is a little bit bigger than like what they're attacking, and may enable them to penetrate rapidly into
Yeah,
deep tissue.
Yeah, that's
So,
the problem, John. Right? Like these things, they just
yeah.
get sequestered in your tissue. They get deposited, like
Yeah.
repositories of, and they make you sick. It's unreal that this is that size issue is another advancement. Yeah, definitely.
So I think like they could be low cost. Like I said, to me, this is one of those things. I read it and it's like, oh, that's great. There's like I can see real potential to this, and
That's
like I
because we're
said,
nerds,
me,
John. We
I'm
can't help it.
fascinated. But I mean, just to talk about to go back to the capillary electrophoresis, one of the ones I worked on was black mamba venom. Like I say, I could buy them from Sigma, dissolve them up in water, and inject them, and just try and figure out what's on there. It was all based on, because the limited tools we had, migration time and then molecular weight. And the paper that I was looking at said there's about 28 peptides in black mamba venom. We actually counted 108 just in the limited range we could pick up. And we couldn't, because we only did 1,200 molecular weight on the mass limit on the mass spectrometer. We couldn't tell if there were higher molecular weight components in that venom.
So John, if I understood you, you were able to characterize it better than Sigma, right? That's what you're saying.
I mean, it wasn't really characterizing. There was no sequential information. It was just purely molecular weight determination, and it was like, say, looking at where they came off on an electrophoregram. Chances are, and there were enough of them that you got two charge states to say this is definitely the molecular weight. Others you only got one charge state because we're right at the limit of what the mass spec could do. I mean, we're really pushing.
Nonetheless, right? The venom of these animals is incredibly diverse.
Oh yeah, absolutely.
Yeah, that's what you're saying.
Yeah. It was like I say, it was for what was just a sort of throwaway idea. As like, oh, I could try this. It was just one of those. The more I did, the more fascinating it was. It was great fun. It was like, it was what science is about to me. You want to enjoy it, and I was like, oh, I have to do more of this. This is brilliant. And then
research, baby. It's where it's at. I love basic. I kind of miss it sometimes, right? That you were doing basic research.
Yeah, and then of course came to the end of my postdoc, and that was it. I went to work for mass spec company in Kratos, who were launching a time of flight mass spec, and they're like, "Well, what can we do to show what this time of flight does?" I guess, hold on, you look at snake venoms with this. Let's see what we can find.
John, I don't know. John, I don't know where you were when you were doing this, but rumor has it there was like a rash of individuals that had pissed you off that suddenly came down with venom. I don't understand.
you know, my wife and I, we love to watch like detective shows and things like that and murder mysteries, and we're thinking, if you like put some snake venom in it, how detectable would it be, etc., etc.
Not very. I'm telling you, especially if I don't even want. This is me being evil. Let's move on. I'm thinking it would be if you were to drop dead in the mall, John. And I don't know if anyone goes to the mall, right? My wife would be mad. I'm talking about people dropping dead. They would never test you for snake venom. That would not be what's tested. I promise you. They would chalk you and me up to old age, right? Or
yeah, and they're certainly not going to look at your Aldrich Sigma buying habits.
All right, let's move. Don't mess with a scientist. That's the moral here. We might not be the most physically gifted people in the world, but man, we know how to get you. No, I'm just kidding.
Exactly, yes. My wife and I both know not to step like the wrong side of each other because we have the ins on this stuff.
Yeah. No, good stuff, John, and great, real fascinating. But I'm excited to talk to Neil a little bit more. Maybe we get one more news article.
This is the with all these, it's always like, what's the one that's the big, potentially huge deal, and this is the big deal. So, first ever gene edit of infant boy leads to apparent cure. So a baby boy born with a debilitating genetic disease will likely no longer require liver transplant to stabilize his condition, thanks to first of its kind genetic medicine he got at 6 months of age. So this was a treatment developed by Philadelphia-based startup iAcure, uses an enzyme to cut DNA at a safe location, then has a virus slip and a new copy of a vital gene. It's the first time that regulators have allowed researchers to edit genes directly inside an infant's body. The CEO of the company, Joseph Truitt, said baby's doing well. Was treated about 6 months ago, and the company plans to present more detailed results at a medical conference in March. The infant was born without a vital enzyme called ornithine transcarbamylase, or ITC. It's the same disease that 25 years ago was at the centre of a patient's death from an experimental treatment that set the target back. Set this field back for a long time. Is that the German thing where it was like 6 patients died? Anyway,
it might have been, but that was a par excel thing. It might be a little bit different than that.
Yeah, I think it might be a different thing. Yeah. But anyway, it affects roughly 100 newborns in the US and 1,000 babies worldwide each year. It leaves patients unable to fully break down protein, causing ammonia to surge in their blood. Left untreated, these protein surges can cause irreversible brain damage. So the only cure is a liver transplant, which can take a year to secure. So they're aiming with this gene therapy to make it easier, and they sort of stabilize the baby boy condition with hemodialysis, strict diet, and the ammonia reducing drug Ravicti, and then he got an infusion of the gene therapy. Within 3 months, blood levels of protein building block called glutamine was reduced, suggesting the therapy was working. So he was starting to be pulled off Ravicti, that's the ammonia reducing drug. And then they sort of said that at 6 months his ammonia and glutamine levels are normal, and he hasn't experienced any more metabolic crises. So I will pass that to you.
Yeah, what a great story, John. I mean, I don't know how much I really need to add just for the sake of time, but like I'm just going to recap it basically. Like this is an incredibly rare, it's like an enzyme disorder, right, John? Rare disease, and to have a setback 25 years ago now, if I read that right.
And it does detail I can give you the details.
What a great CEO though. This guy, he kept at it, didn't he? Here, 25 years later, even though I think that inadvertently maybe killed somebody, that didn't stop him. And here we are 25 years later, and they're saving lives, John. This is unreal. So this is a bioanalytical show, right? And so the glutamate's important. A lot of enzyme replacements, that's what this falls into, that are rare diseases. One of the best biomarkers is the measure of glycogen, which is, I know, not glutamate, but it's similar. And so this becomes a very powerful biomarker. And that's the first sign that this took. I bet you the physicians were shocked, and it's an incredibly promising thing. And as they talk about this, they take him off of another medication. This child potentially could have a normal life. Unbelievable, John. Think about, I don't know if, I think of myself as somebody who, when I get knocked down, I get right back up. But man, I don't know if I would have spent 25 years still fighting for something. It's an unbelievable story. I think in and of itself, the CEO is heroic. But then there's some haters out there, right, John? The article talks about that too. That's the interesting part about this, because it's not CRISPR, it's this ARCUS technology, which I hadn't really heard of that until this article. So just it's great. We could probably digest this article a lot further. I don't know if you have any closing remarks, but wow, really, these unmet need, there's only like 1,000 of these disorders, and these things tend to get left behind. And it's great. Remember, I feel like post-COVID, right? There was this, right before COVID, John, we had a lot of interest, and everything was kind of diversified from immuno-oncology. And then I think we kind of elasted back right into immuno-oncology. And now I think we're getting back into the more rare diseases. So it's great stuff, John. I love it. It's wonderful story. Feel for any families that have these sorts of disorders, and now there's hope, right, John? Because I think it doesn't end here. I think it can be applicable to like Pompe disease and all these other rare diseases they're rare, but there's certainly a little bit more number of instances than what this disease has. So great stuff, John. Really cool stuff.
Yeah, absolutely. So I don't really have much to add to it. I just thought it was a fascinating thing, and you're right. There's on so many levels. this could be a path forward for a lot of
They,
other
they
stuff.
think 2027, right? They think they might have this to market by 2027. It's really revolutionary stuff. It's to me, it's like almost out of a I don't know sci-fi movie, right? Where you
There
can
we go.
perform one
That's the
injection, and it's just unreal.
Yeah, and think how many times you say that in the course of this podcast, because it's just. But what the reason why I highlight that is because we read so many things like this, and just how quickly medicine is potentially moving. Everything. So much today is like these are new things that are steps forward that have potential for the long term for other conditions and could really make a difference.
Yeah, awesome stuff. Well, with that, John, right? Are we wrapping up the news and notes?
I
And
think so.
I'm
Yeah.
sure Neil is anxious to jump right in because I think we've got a real fascinating topic. Why don't you tell us a little bit more about what we're talking about today, Joe?
So I'm just going to give a headline, then we'll get a chance to introduce Neil and let him introduce himself properly. So the background to this is both Dominic and I were at EBF in November in Barcelona, and there was a session on patient-centric sampling, which was honestly fascinating. And as a bioanalytical lab, we're sort of getting involved in it. It's sort of relatively early days for us in terms of how much work we've been doing in terms of working with some of these devices. Although some of us have history with dry blood spot in the past. And I thought it would be really nice we haven't touched on it at all on the podcast, so it seemed an appropriate time to talk about, as we put here, patient-centric sampling, expanding sampling beyond the clinic, and the potential logistical and bioanalytical challenges. Like I say, we're neophytes in this, but Neil is much more of an expert in this. He is an expert in this field, and I thought it'd be good to have someone on to talk through this and say, because this is where bioanalytical is going in the future, and it has been talked about for quite a while. But we routinely talk to customers, and this is why we haven't really done much to date, because it's where we go with the science is customer driven. But we're working with some people already, and then we just had a conversation yesterday talking about that there's more potential to do more of this work from now onwards. So it will become increasingly part of the conversation. So, like I say, the timing just seemed right to have a guest on to talk through patient-centric sampling and what it can mean for the industry. So with no further ado, I'd like to introduce Dr. Neil Spooner. And Neil, would you like to say hello, introduce yourself, and give us your background and how you got here today?
Thanks, John. Thanks, Dominic. Thanks for having me today. I'm really excited for this. So yeah, I'm Neil Spooner. I'm just really looking forward to having a chat with you today on this podcast.
So do you want to give us a bit? So obviously, I've known you for years. I knew you from your GSK days. Do you want to talk us through, your career with this? Because obviously, I know there was the big drive at GSK in terms of dry blood spot sampling, and I assume, like, when I talk about the snake venom and how excited I get, I assume that's what got you into this realm.
Yeah.
And how you're excited about it going forward. Just tell us all about it.
So my background is bioanalysis. I did a postdoc with thermospray. So if we're going back in history like you were, John, I did some thermospray stuff back in Bristol when I was doing my postdoc. And
so was that with Jeff Eglinton?
Yeah. In that
Okay.
group.
So what year were you there, Neil?
I was doing a postdoc looking at chlorophyll degradation to see how that turned into porphyrins in rock.
Sure. And so what was your timing at Bristol?
Oh, I can't remember years, John.
Oh, okay. I was there.
I'm coming up for 60, so a long time ago.
I may have predated you then, because I was there at 84 to 85 doing a master's in analytical chemistry, and that was the light bulb moment for me. That then I loved chromatography because luckily at the end of the masters you did like a project, and I ended up doing a GC project for a compound called oxalinic acid. Should have been done by LC, but we had the limited supply of LCs, Sad to do it, but GC. And then finally, towards the end of our project, I went out to a local water company who had the GCMS, and they ran a couple of my samples by GCMS, and that was when I saw what mass spec could do, and it was like,
Yeah.
oh, this is it, chromatography and mass spec, whoa! And that was it. But that was Bristol basically set me on that path. So I was like, where the Eglinton group were probably on the third floor of the chemistry department, we were on the second floor in the masters group. So anyway, I'll shut up because that's irrelevant.
No problem, John. But from there, I went to work for what was Huntingdon Life Sciences at the time. It's now, that site's part of LabCorp now, for a couple of years, and got my hands on a Sykes API3 for the first time there. And then got a job at SmithKline Beecham, which morphed into GlaxoSmithKline. So I had 20 years at GSK, mostly in the UK, but did 3 years on the outskirts of Philadelphia as well. And in my later years there was running a group looking at new technologies for bioanalytical workflows. And at the time we got interested in, we knew we'd got a need to develop some of our drugs with trials in children. And we also knew we were taking far too much blood from mice and rats. So we were starting to poke around at microsampling approaches. And that's where we got interested in dry blood spots. And so at GSK, we were the first pharma company to bring that kind of approach into our clinical trials, and also some alternative approaches for non-clinical microsampling. And through the group, we figured out the benefits of being able to collect smaller samples, potentially in people's homes, stable samples at room temperature, being able to ship them to the analytical lab. But there was obviously the problem around the quality of the data that came out of traditional dry blood spots. So started to encourage companies to develop the next generation of samplers that would collect, that would have all the advantages of the small volume, the dried sample, the room temperature shipment, the home collection, etc., but that could collect an accurate volume simply. So, it was working with James Rudge at Phenomenex at the time, who got the idea for the meter tip and brought in prototypes into our lab at the time, and helped him test those with a guy called Phil Deniff. And he was able to get enough data to sell that to his management, and they did a spin out company, and that turned into Mitra that is available now through Trajan, and started to lead to a plethora of different technologies. So about 9 years ago, in their wisdom, when GSK had yet another reorg, and my job didn't exist anymore, I got the opportunity to take redundancy and set up my own bioanalytical consultancy with the main focus on these patient-centric sampling approaches, working with pharma companies, CROs, technology companies, central labs, whoever, and just helping them develop the technologies, understand what the technologies were, what they weren't, and moving that forwards. And with time, we, at a conference just outside Philly about 7 or 8 years ago, there's a small group of us in a room, people from different backgrounds. There were pharma, central labs, CROs, clinicians, academics, and we all realized that getting these technologies routinely accepted, whether for clinical trials or healthcare or drug testing in sport or therapeutic drug monitoring or consumer-led health, was going to be hard. We were in the middle of the Theranos mess. And so, there was that whole thing that people who said you could do things with small samples weren't being believed because Theranos had lied and made stuff up. And so we decided to form an informal grouping of us that we called the Patient-centric Sampling Interest Group, the idea of which was to work across organizational boundaries to just help promote the use of these technologies, the broader understanding of them by different user groups, figuring out what the technology, what the challenges were with the technology, and then could we do things to overcome that? And that built up and got bigger. And so in March 2023, we turned it from a loose amalgamation of passionate people that wanted to push it forward, keeping that ethos, but turned it into an actual not-for-profit organization that was able to be all grown up and have its own bank account and raise money through sponsorship, and therefore start paying to do a lot more activities. And that meant I could spend a lot more of my time doing that. So that's what I spend most of my time doing now. The consultancy, I do a small amount. I have to be very careful with the consultancy because I can't consult with people who are members of the patient-centric sampling interest group, because I can't be seen to be showing a bias. So my consultancy has to be in other areas of bioanalysis now, with some interesting, mass spec interfaces and things like that are in the research stages. But, the patient-centric sampling interest group is going from strength to strength, and I'm the chair of that. We've got a board of 8 of us, and we've got a bunch of activities that, we can talk about later. But that's my story in a nutshell.
So how many member companies or group, I suppose companies is the wrong word, but organizations do you now have in that interest group?
We base ourselves on the passion of the individual. So we've got, so on our LinkedIn group, which is where we talk to each other, we've got just over 500 members of that now. Wow. And a pretty active way of talking to each other. On LinkedIn, we also have a company, which is how we talk to the external world. But we have, I think it's 21, 22 sponsors. If you look on our homepage, our website, we set the annual fee that the sponsors pay is low. It's £2,500. So it's affordable by small companies and large companies. So the idea is to have a large, diverse group of sponsors rather than 1 or 2 big sponsors paying a large amount. It doesn't matter whether you're large or small, you pay the same amount. Individual members don't pay anything. They're free, because we looked it up and it was just going to be too much hassle to chase 500 people every year for a few dollars. And we'd built ourselves from the passion of individuals giving up their time for free. And once you start throwing money into that relationship and asking people for a few dollars, it kind of changes that. So, no, it's been very, very successful. And,
so I want to drill in for just this is a microsampling group, or what exactly is the sorry? Maybe
so it's the patient-centric sampling interest group. So
okay, great. Well, we'd love to learn more.
Focused on the clinical side.
Yeah. But using Metro devices and Tasso devices and things like that, or okay.
So maybe Neil, it might be good to just say define what you see as patient-centric sampling, and then why there's moving in this direction, and I think that's all very helpful to just talk about.
So it's not about microsampling. The samples could be larger. It's about collecting the sample in a way that puts the patient at the center. So traditionally, when we collect blood samples, whether for clinical trials, for healthcare, drug testing in sport, whatever, we use the standard tubes that take a large volume, a couple of mils or whatever, and it's designed to be collected in that way for the needs of anyone but the patient. The patient comes last. So it's the needs of the analytical lab, although the analytical lab doesn't need that volume anymore. They throw most of it away, which is not ethical.
Yeah.
It's the needs of the doctors, the phlebotomists, the shipping. It doesn't think about the patient. And what we're trying to do with these technologies is turn that on the head and say, well, it's the patient that we're taking the sample from. Shouldn't they be the center of what we're thinking about here? Why are we taking so much blood from ill people and throwing it in the trash? That's not ethical to me. We can work with a lot less. We can, with these technologies, whether it's, there's a variety of them that can collect things from your finger prick or from your upper arm. They can collect liquid samples. They can collect dried samples. They can collect plasma. They can collect whole blood. And also, patient-centric sampling is about other biological samples, urine, feces, and things as well. But the main focus is on blood.
Mm-hmm.
A lot of exciting things happening with urine as well, because just peeing in a pot is very messy, and there's better
Mhm.
ways of doing that. But if we put the patient at the center, what can we do? At the moment, our clinical trials are restricted to people that can make it into a clinical center. If not, they've got a long drive or a plane ride to get to a clinical center. It's expensive, it's time consuming, it restricts recruitment, healthcare.
Hard to adhere to it, right? Like in general, you miss a day, right? There's all sorts of complications. Yeah. And sorry to jump in on you, Neil. I hadn't really thought when I hear patient-centric focus, I hadn't really thought of just how dated we are. So you're right. I guess this is like eye popping for me. We've been collecting blood the same way we have since World War II, essentially, right? And we've made all these great advances in immunoanalyzers and all sorts of, but it's never made its way to a patient, has it?
Yeah.
That's fascinating.
We're sticking
All
a
right.
needle in. There's a lot of people that experience pain.
I do. I hate. I'm one of them. I can't stand it. Everybody's like, I'm kind of wimpy. You stick a needle in my arm, and I don't care how small it is. It doesn't matter. You stick something in me, I'm pissed. I'm telling you, I don't like it.
There's groups out there. We had a webinar just before Christmas in a remote area in the UK near the Lake District, where they've got a bunch of people with special educational needs that because of their needs, it's difficult to sample them. All of them hadn't had a blood sample for at least 5 years. Some have never had a blood sample taken.
Well, because they just couldn't. They're just too antsy, or right. There's
They
no.
bring these technologies in, and suddenly they're able to get blood samples from them. Suddenly they can start giving them proper healthcare. They can start doing diagnoses. There's all sorts of places where this can apply. And, whether it's healthcare or whatever, if you can do it in the home and send it off, not only can you think about, okay, the samples we're collecting now, could we get richer data sets for our clinical trials? Could we get richer data sets for healthcare? Could we be monitoring people earlier in life before they get ill? Could we be monitoring people more regularly? And
Well, Neil, I think the answer, like, it's a resounding yes. I mean, I've only now realized, because I have like impacted veins and they're kind of deep, and just I'm a big guy, right? I'm a pretty muscular guy, and you would think that they'd be out. They're not. You got to turn my arm. You got to get to a real sensitive part of my arm. I pass out sometimes if the phlebotomist doesn't know what they're doing. And so lately, it's just been coming out of I just go right to my fist, and that's how I give blood now. But it does dry up a little bit. But Neil, you're spot on. This could, if you could simply do it at home, painlessly, I would send a wellness sample in every 6 months if that's what they were asking for, right?
And it doesn't even need to be at home. It could be at your local pharmacist. It could be at your doctor's.
Understandably, just as long as I don't have that anxiety of going and getting a needle stuck in and wondering if I got a nurse or a phlebotomist that knows what they're doing, because I have to ask. I've had most of them. They're like, "What? Okay, let me go get someone else." Right? Because I'm gonna there's a 300-pound guy about ready to fall on you if you draw blood from me the wrong way.
Yeah. And a lot of this is just about giving patients choice. Some of them might want to go into the clinic and have it done in the traditional way. Some might not. And if we can give people choice, that's what it's about. It's put the patient.
Neil, you're a fascinating individual, Neil. I'm just getting to know you. You've had this unbelievable great career, and you could have gone off into the consulting world like some of the other people I know and done well for yourself financially. Instead, you got this altruistic side to you. Good for you. You've taken things like, what you're great at, you want to give away. So I applaud you for that, Neil. So it's wonderful that what you're doing, because this is really intense stuff you're talking. To me, this consortium you guys are forming could really revolutionize patient-centric testing, or even what we call personalized medicine, right? Those are big words. They're somewhat, to me, they can be interpreted different. So Neil, I did not know this about you. What a fascinating turn you've made, because this is within the last 5 years, or how long have you been consulting?
So I've been consulting for 9 years, but this being full-time patient-centric sampling interest group has been, I guess, the last year and a half.
Wow. Well, we're going to sign up. We want to. I have a couple colleagues that might be of interest, but I want to get back on our agenda, John. Bring us around. So we went over whys, right? Do we want to talk about dried blood?
Yeah, I think it's worth touching on the history, because again, it's like dry blood spot sampling, this is where I think of it. I mean, certainly in terms of, I think we had initial discussions because some of the drugs that we were working on for customers were going to be administered in the third world. And so it was a case of, was dry blood spot a way around that, where you could, obviously take the sample onto a card, ship it at room temperature, and then analyze it at the lab. And actually, when I ran the lab in the Netherlands, they were already supporting a couple of clinical trials with dry blood spots. And they got through validation, and so there I was seeing the practical implications of dry blood spots, where it not. And I hope, Neil, you talk about hematocrit, etc. This was more just the practical challenges of processing cards with thousands of samples, and it was very interesting from that perspective. So that's why I think it's worth touching on it. I know it's all moved away from it, but I think as part of the history, it's a really good topic to touch on.
Before Neil jumps in, we may disagree on some of this, because remember, we're a bioanalytical show, right? So I'd like to make sure we drill into dried blood spots, in my opinion, on paper look fascinating and such good utility. And then I'm not a small molecule guy, so
I'll be honest. I remember hearing some of Neil's talks at, I don't know what conference, but talking about dry blood spots and thinking, holy shit, this is great. I think this is where we're going.
But
let's hand it back to the expert.
Neil, talk to me a little bit about these things, because those devices I mentioned earlier, maybe again, I want to hear your side of things, but you can get a sense that I'm not as bullish on them as others may be, and you haven't really shared your thoughts yet. But please tell us a little bit more about the utility. And we are a bioanalytical show, right? So that's to me where we want to keep the utility of it lies in there for us, rather than I do appreciate the patient, but please tell us about it.
So obviously, traditional dry blood spots had a lot of advantages, say small volumes, room temperature shipment, but there were some problems. Obviously, they work fine for neonatal screening and a lot of applications. And you have to think, what is the quality of what's the amount of variability you're willing to have in your data that you're willing to accept? But for a lot of things we were wanting to do with drug development, it wasn't enough. Plus the fact it's a very different format of sample that's trickier for the bioanalytical lab. Although there was some automation starting to come along to automate the processing and analysis of these samples, so you could do them online, elutions and things like that started to appear.
Yeah, if I could just jump in, the lab in Oss, we had an automated puncher for the cards, but it was actually from a company in Australia, and they were no size. So when there was a problem with the instrument, it went down for months at a time because the technical support wasn't there.
Yeah, so I want to make sure our audience grasps this. You're going back 25. This is not new technology. Dry blood spots a long time. So just to set the stage, you're going back to having some automation with these things. And they had applicant, when I say these things, dry blood spots, you're going back 20 years, 25 plus years, right? But it's not a mainstream thing, right? It really isn't.
Yeah, no, it's not. But so that's why we started to develop these next generation technologies that could collect a sample that was an accurate volume, getting past the hematocrit problem, that was in a format that was maybe more amenable to automation. And some of the technologies collect liquid samples that people are starting to demonstrate. So long as you can get that to a laboratory within a couple of days and centrifuge it and store it in the laboratory, you can kind of change the workflows a little bit. So we're on kind of second generation of these technologies, and we'll start to see the next generation of technologies. We're starting to see bubbling under in academia a next generation of these technologies. So, I look at it that at the moment we're like at the Nokia phone, which was really a perfect mobile phone, good battery life, easy to use, all these great things. And then the iPhone came along and blew it out of the water. And no one talks about Nokias anymore, apart from maybe their elderly relatives. And that's what we've got coming over the horizon in the next few years, is the iPhone next step that will help collect these samples in a patient-centric manner and potentially can start some of the analytical work at the point of collection in the device. So that then changes what we do in the lab and how we do the analysis and where the analysis is. And if people can be brave enough and imagine a different future that doesn't rely on a wet sample having to be shipped around the world and stored and analyzed in the laboratory and then moved to a mass spectrometer or an immunoassay platform, and we can do some of that differently, and we can do some of that pre-analytic work and some of that sample prep at the point of collection as part of the collection device, then that prepared sample go onto an analytical platform, we get to a very different place. And I think we'll start to see that in the next 5 to 10 years.
Amen, Neil. So I want to make sure, because you're a small, you guys, John and you are small molecule, right, on biologics.
Sure. Yep.
So part of that, and I love what you're saying, that means somewhere in the, whatever the collection device may be, you've got like something to stabilize a metabolite, maybe help you with your elaborate a little bit on what would be some of the engineering, right? This is engineering, in my opinion.
Yeah, you can start building in stabilizing agents. You can start building in capture antibodies.
Yeah.
You can start building enzymics into there. You can start building a kind of solid phase extraction into there. It can all be done with a sample when it's still liquid, collected by a human being, and while they're washing their hands and dealing with things, it's doing stuff, in the same way, think of the lateral flow test we were all using during COVID. You develop it, you're adding a liquid. Everyone said this kind of stuff couldn't be done. We got to COVID and suddenly we realized in order for people to go to work and kids to go to school, we needed to get over ourselves. And suddenly people were, taking swabs, putting it into a buffer solution, adding it to a lateral flow device and making a reading. And that was done by billions of people a day.
Yeah, some better than others, but yeah, right. Some were more reliable for sure, the accuracy of them. But no, you're right. I preach it. It's funny how in an emergency, right, or some sort of kind of catastrophic event, science has rose up to meet this challenge. And then, you're right. Why aren't we using this across the board, right? That's kind of what why, if we did it for COVID to make this advancement and being able to do testing at home to see if you're positive, why can't we do it for a whole multitude of stuff, right? Is that kind of I just want to make sure that's me thinking, not, please clarify it. Yeah.
And some of them, the diagnostic might be able to be done at home, but some of them you're still going to need to send it off.
Sure.
But that sending it off to somewhere, that could be, you think of the old days of photography with film, and you used to take it to your local pharmacist or chemist, or as we call it in the UK, Boots, to get that developed. And developing a color film was quite a complicated process. But they built machines that were good enough to give you printed color photos that a teenager working their weekend job was able to run that machine. Didn't need anyone with expertise. And you could take it to your local chemist and they could do it there. So there's a potential for some of our diagnostics to be done in a different place and just turn, they don't necessarily need to go to a traditional lab. Yes.
You're a revolutionist. You want to revolutionize medicine, Neil. You're like a rebel. You're a maverick.
That's what Theranos were talking about. And a lot of what Theranos talked about made sense.
But it's not the industry back though, right? You're bringing up somebody, not, I don't know if, right? That set this field back a decade though, Neil. You can agree to that.
Absolutely.
Yeah.
That is why we formed the Patient-centric Sampling Interest Group, because Theranos had set us back a decade because of their lies.
Yeah. What a great story that is. Oh, I mean, great, horrific story rather, but geez, yeah. You're right though. Sorry to interrupt you there. You're 100% right. That is why that field almost got killed. because of her that whole, let's just call it Ponzi scheme they were running. And now you're revisiting it. Good for you, Neil. This is amazing. I want to join your group now.
You're more than welcome, Dominic.
Do we? I know we're all over the place. Do you want to talk about some of the devices that are currently out there, Neil, just to give like,
I don't want to get into naming them.
Oh, that's fine. That's not a problem.
All right.
There's devices for finger pricks and upper arms.
Yep, that's fine.
You can see them on our website. On the PCSIG website, we've got a comprehensive list of most of the technologies that are out there, and you can get a link to.
Mad respect, Neil. We can move on. Great respect.
That's absolutely fine. Yeah, and we can cut this bit if it works better to do that.
But I do want to talk about the next item on our agenda, John. Right? I hope we're going to dive into how we validate these things.
Feel free. Yeah. Go take it wherever you want.
Yeah. So Neil, I think that's where I'm kind of leading up to. I know we're over the hour mark here, but I hope we can do at least a good deep dive into the validation of these things and. Just tell us about, like, to me, well, why don't you tell us like how much more challenging, maybe, I don't want to bias it, right? I'm trying to phrase the question without being biased. So are there bioanalytical validation differences from a plasma to a microsampling?
I think it's how does it change your approach in validation?
Thank you, John. Yep.
And the answer is yes. And what we need to think about is, why do we validate an assay? Because a lot of people forget why we validate an assay. The great thing about guidances is they give us some rules. The bad thing is it stops people thinking. And what is validation all about? And it's because when we're doing bioanalysis, a lot of people, when I ask them what is bioanalysis, they start talking about measuring the concentration in a sample. And immediately they are wrong.
Yeah, it's about verifying the analyst, the instrument, and the method, right? Like, I think of it as, I don't know where you're going, but to me, when I hear the word validation, you're only verifying that you got reproducibility of the analyst, the instrumentation, right? The process necessarily. But maybe I'm wrong. You please answer it. What is your answer to that?
But people forget that what they're measuring is not the concentration in the tube. They're measuring the concentration in the human being or the animal.
Yes.
And people always forget that. And it's like, come on, what are you doing here? You're developing a medicine for people. You're making a measurement for clinical diagnostics for people. We're not curing tubes. We're not making medicines for tubes. We're making medicines for people. So validation is about understanding what happened to the thing you're trying to measure between the moment it left that human being or that animal to the moment you generate a data point.
Yes, I agree.
And getting people to understand that. So thinking through what on earth can happen to that sample during that journey, and what's different with a patient-centric sample to that. So if you're wanting to ship it at room temperature, you've got to demonstrate that you've got stability at room temperature. Simple as that. And if it is at room temperature, ambient temperature, what does that mean? So as your sample's moving around the world and it's in the belly of an aircraft, or it's on a FedEx truck on a runway, or it's being dumped in the corner of a room somewhere, what does ambient temperature actually mean? And sometimes it might be 40 degrees Celsius, and sometimes it might be minus 20 degrees Celsius. So your validation has to take all that into account. And so you've got to understand your molecule in the matrix that you're interested in, and is it stable?
Yeah.
And that then builds into, how can you ship that sample around the world? You may have had the dream of being able to ship it at ambient, but you then find, when it's on this FedEx truck at 40 degrees.
Yeah. Unless you spend incredible amounts of investment into temp tails and specialized packaging, Neil. But you're right, the standard shipment of a blood sample does not entail what I just said there. So there is huge fluctuations in temperatures. It's a good point. Please continue.
It can be. So there's a whole bunch of companies now looking at that logistics bit. So going off topic a little bit, there's companies looking at the logistics piece. Can they stabilize that sample at whatever temperature you want to put it in there at? Can they track where that sample's been? Can they track how much it's been shaken about by someone? We don't do that at the moment. A lot of frozen samples. Are we really sure your frozen sample did stay frozen for the 6 months until you analyzed it?
Yeah.
Sometimes during transit, we don't know.
Yeah.
And,
Even
and
the collection tube itself can be
we have to be
challenging, right?
careful.
Yeah.
We don't put extra rules on this new sample type that we're not putting on our existing sampling type.
Okay. I hear you there. I get you. That's a valid point. There's much more scrutiny if a dry blood spot is to be validated versus a K2 EDTA sample or a plasma sample where no one, same tube, all good, right? That's interesting, Neil. Great point there.
And so, but you need to validate that kind of thing. You need, hematocrit, the amount of red blood cells in your sample. For dried samples, that can affect the extraction efficiency for some analytes. So you need to investigate that. You just need to look at things. We discovered when we were doing some work on pediatrics with dry blood spots, the way, by sitting down and talking to a pediatrician clinician, the way that they collected sample from newborn babies for a heel prick to get enough, these are really tiny preemies, to get enough blood and it not drip off everywhere. They make a well of paraffin wax, and we call it Vaseline, and the blood accumulates in there, and then they're able to transfer that onto the dry blood spot cards. We didn't know that until we asked people.
Sure.
And so you then have to look, okay, what the hell does paraffin wax do to my dry blood spot card?
What's your absorption? What the hell's going on there? Yeah, okay.
That would wreak havoc on when you go to do a bioanalytical test if you haven't accounted for that. Neil, this is really cool.
And how much of this, so with traditional blood samples, and you're running a clinical trial and you're in 100 different centers all across the world, and you're now in phase 3, so you're in patients. So the nurses and doctors don't really give a damn about your clinical trial. They give a damn about the patient. And the patient happens to be on your clinical trial, but they're taking all sorts of bloods. They're doing all sorts of care with them. And they happen to take your blood for your precious pharmacokinetic measurement. Do you really think they're getting it into the centrifuge and on ice and into the freezer in the timescale you think they are? No, they're not. It's not their priority. Their priority is the patient. But no one's ever opened that Pandora's box and asked that question. But we ask it of dry blood spots and microsampling and patient-centric sampling. And I challenge people to go to clinical centers, because I think this is a really important thing that bioanalysts should do, and that is talk to their customer. Go into the clinic, go into the animal room, find out what on earth is going on there, and it's an eye-opener.
Sure.
And you may have wonderful things in your study reference manual and your protocol.
Yeah.
That ain't what's happening in the field.
Yeah. I don't know about you, John, but what he just said, I've done a little bit of, not maybe to the degree, Neil, you're talking about, but like when I was trying to stabilize bone marrow aspirates for flow cytometry, and this was back when I was at Streck, it was up in Omaha, and I went right into Creighton University and sat in the surgical room and watched them draw bone marrow aspirates from like 3 patients that we were going to use, try to stabilize it. We're trying to stabilize it at room temperature for like 7 days. So that the very thing we're kind of talking about. Man, I don't ever want to get a bone marrow aspirate, but I learned a tremendous amount about the plug, about how they're drawing it, and how we had to take that into account when we went to stabilize it, the impact, underdraws, overdraws. So, Neil, I'm with you. All good scientists should go and try. Hey, you can read it, but man, you got to go touch it to truly, and even interview it, Neil. It's really good. I'm like I said, impressed. Maybe I'm easily impressed. Maybe some of what you said, I think people do naturally, but to be doing it proactively is really interesting. And I do think now that you're talking to me, I'm like, this 3 phase 1 units here, I should go hang out at them. Right? That's what you're saying, right? We got plenty of opportunity. So, and I have visited a few of them, but that was a little bit of a sidebar, Neil. But I think every bioanalyst out there should really hear what you just said. Hey, don't just hide behind the desk here and hide behind the biological safety cabinet or wherever you're in. Hey, go see it. Go learn more about what that physician and clinician's doing, Neil. That is really important.
And I'll be honest, I've seen the facility, I've toured facilities, but never actually seen them actively taking anything from patients. So you get the setting, but that's only a fraction of the picture there. So I totally hear you. That's really, yeah.
And to your point, Neil, it was a pathologist, it was an MD who was doing the bone marrow aspirates. So we did interview that. I sat down with him afterwards and we talked about things to kind of what you're getting at, so that we could design a proper way to 510K clear it. That's what this was under. But Neil, it's really, I hope if there's any young people out there, or even, hey John, even you, it's never too late, right? But this is
I agree. Yeah,
really
yeah.
important.
Go connect with the patient, or if you're doing animal studies, go spend a day in the animal house and just watch when the pharmacokineticist is asking for 3 samples in the first 5 minutes after an IV administration. You just go in there and watch those poor people and see what they have to go through, because
Sure.
you want your perfect PK curve.
Or hey, collect on wet, dry ice or whatever, right? It's not that easy.
Yeah.
There's some technique there.
So obviously we're running quite long. We have a couple of other topics that I don't know how much you want to say on either of these. I think probably the one I would really like to know is how involved are the regulators in this, Neil? Are they well engaged with your group, or what's the deal there?
Regulators, if we're talking about pharmaceutical development, are really, really supportive. There's particularly in the FDA.
Yep.
Particularly if you go talk to Brian Booth at the FDA, who's been key in our bioanalytical world for a long time now, and he's given quite a few talks. Some of them are recorded on our website, where he's given talks, and the FDA are very, very open to this technology. They want you to do good science. They want you to think about your validation. They want to think about your sample transport. They want to think about the things that could get in the way of you generating good quality data. But if you thought about that and you've done the right validation and you've put the right things in place, they're really open about it. They're really, Brian is saying, go do this. This is where this brings benefit to your trial, to your patients, if it's going to help with recruitment and retention, if it's going to give a better experience to the patients, go do this. There's no blockers from the regulators. Now, when you get to diagnostics, particularly in the US, it gets more complicated, because home testing, lab developed tests, there's a lot of stuff going on there between the FDA wanting to get into regulating this stuff and other bodies wanting, and it's getting complicated with diagnostics. Yeah. So we're looking, can we bring together diagnostics companies and that part of the FDA, and maybe other regulators as well, into the same room in the same way we did with Crystal City 25 years ago. And then that series of, what was it? How many meetings over the last 25 years? 8 meetings that resulted in ICH M10. But back 25 years ago, validation for bioanalysis was the Wild West.
Oh, it was all over the place. Yeah, absolutely.
There were no rules. So we brought, people brought together the regulators and pharma and CROs, etc., into the same room to talk this through, to start reaching consensus. And over the years, we've reached consensus. We can do the same thing for diagnostics, for lab-developed tests, for home sampling.
Neil, I think you're winning me over here a little bit. Meaning, I'm a little bit. I'm not anti these things. I just think they're overhyped. When I say these things, I mean, in general, a lot of microsampling, or maybe even some of the kind of patient-centric sampling you're talking about, people, there's usually a sacrifice in the limits of detection, right? I'm just going to jump right in. And those, but I had always taken it from the wrong lens. I'm looking at it for my needs and my organization's needs, and that's a mistake. And I like to think I don't make that mistake too often, because it should be taken from forget the bioanalytical method, Dom. You're way over here trying to do PK and all this stuff. Hey, how about just simple diagnostic tests and standard tests. And yes, maybe I think it has tremendous utility in PK, especially if it's like you touched on it, pediatrics. I mean, these are definitely, absolutely, that I wouldn't, doesn't matter what result you can get. If you can get any result at all is impressive
Absolutely.
because
And
of the volumes.
honestly, how often do you need the 2 mils of blood?
Almost never. Yeah, like only if I'm doing a PBMC test or some sort of, and even like I'd have to isolate PBMCs. Otherwise, we don't need much at all, right? So that would be the only time. To your
And
point,
some of
right? A
these
flow test,
technologies,
basically.
and that's why I say don't mistake this for microsampling. Some of it is
Yeah,
microsampling, but some of these technologies can get half a mil easily. Some of them can get towards a mil. So
you can still use a traditional bioanalytically validated method instead of trying to do, thank you, Neil. Instead of trying to do, so that's an uphill battle because I like to think I'm an open-minded guy, and it's hard to really grasp some of the because I think not a lot of us, but those that have tried to validate these things, they tend to just kind of fall short, right? You get a lot of BLQs, right? That's often, I can't speak so much to small molecules, but often in large molecule space, you just simply can't detect it because you're taking such a small, to your point,
people
you're
are
taking
really,
such a small amount. And then it's really, they've had some success with immunogenicity. We have. But again, it's the sensitivity is just not there. So, but that's not what you're here to talk about today. And I think people, when they hear this, especially, I consider myself somewhat knowledgeable about this, right? I go right the wrong way. And you're right. The focus needs to shift towards, hey, the patient and what the needs are there. It's great. Yeah.
but you
I
can
think
still get your sensitivity. You got to just look at what the analytes you're wanting to measure, what the patient group is, what the needs are of your clinical trial.
And Neil, if I hear you right, you can customize. Sorry, I'm talking over you, but if I heard you right, you can customize the way you're collecting that to your needs. That's the power.
There's over 50 of these technologies out there commercially available. There's going to be one that can work for you, and a lot of them will collect larger volume liquid samples that you can work with. And whether you're shipping it as a whole blood sample and it's been centrifuged later, or there's a bunch of the companies out there that have developed now home centrifuges, battery operated, reusable, user-friendly home.
Why not? I can pump my tire up and blow my sprinklers out. Why can't I? Why not be able to do a centrifuge, right? Like, why not?
Yep. So, it's and that's what we need to be working on. Where can the technologies as we have them be used, but where it doesn't work, but we would like to use it, what are the barriers? And that's kind of what PCSIG does all the time. We do a lot of education. We do a lot of going into places and saying, okay, you're saying you're interested, but you can't make it work. What can we do to make it work? What do we need to do to work together to make this happen? Rather than just saying, it can't work and walking away, let's do something about it. Let's figure this out together.
Yeah, I think we could sit here for another hour and 20 minutes and talk more, Neil, but I know we all got maybe time restraints. Why don't we start to just wrap up? If you have, John, if you got any last thoughts, I certainly don't have any. Neil's got to get in the hot seat here real fast.
Yeah, I have no final thoughts, but I really thank you so much for this, Neil, because I came with some preconceived notions. It's really interesting to get background thought processes and everything else. And
so did I, John. I was coming to pick a fight with Neil when I heard, devices, this, and I realized, wait a minute, there's no fighting here. Dom, cut it out. I'm not. Start looking the other way. It's great, Neil. I echo what John just said. Really,
like you said, this was a fascinating session at EBF, and I really wanted to hear more, particularly as we're starting to get involved in it as a bioanalytical lab.
Yeah.
So I really appreciate your time on this.
Well, just something that I want to say as well is the PCSIG, as a bunch of volunteers, we ran a clinical trial that's just getting its readouts now to understand whether home versus in-clinic sampling increases recruitment and retention. And we're getting really good data about patient preferences for home sampling, small sampling, being willing to be sampled more regularly. We're getting really good data showing that more people consented with home sampling versus in-clinic sampling. We're getting really good data to show that recruitment rates are faster. So we increase the speed of clinical trials. We can take months off clinical trials. We can keep patients on clinical trials using these kind of technologies.
Sure.
That saves millions. It takes months off drug development. It's phenomenal when you think about it in other ways as well.
Yeah, you got me almost speechless, Neil. You're right. This has tremendous time savings, which translates into money savings. You're right. And not only that, I think the quality of the data, you mentioned even more sampling, all of that can really increase the probability of a drug's success too, right? Like just understanding, just how the pharmacokinetics of it, the efficacy of it by more but more precise sampling, right? But this has been a wonderful.
We're able to monitor our patients more often in population PK. We know whether they're on a clinical trial or whether they're sick people taking their medicine. Most people don't take their medicine as they're supposed to. If we're able to monitor them more often during a clinical trial for population PK, we get a better idea. How many good drugs have we thrown out because people weren't really taking medicines?
And to your point, Neil, maybe you got to take it every morning. And, if you take it in the afternoon or evening, that could kind of screw up the kinetic of it, right? So it's got all sorts of things to it that are this is just great. But for the sake of time,
we got to wrap up.
Let's get you in the hot seat real fast. So I'm going to kick us off with the first question for you. Tell us about your favorite dish. What's the favorite dish you like to make?
Well, it depends. It's cold and wet out there at the moment. So things like a curry or a roast is good. If the sun's shining more, barbecue.
Okay, well, let's talk about the curry there. You elaborate a little bit. Do you like the yellow curry, red curry? What's your...
Indian curry.
Indian curry, Thai curry, just those rich, spicy flavors and just the fun of making it,
Oh, yeah, it is do you like it to the point where your head's sweating and stuff, or you like it hot? You like it Thai hot or anything, Indian hot, or how hot?
I like flavorsome, but not blow my ears off hot.
Yeah, I'm kind of. I like a little. I definitely want to know that I've been eating spicy food. I don't mind a little sweating at the top of my head. And my wife, when we go out to eat, sometimes like, don't do it. I'm like, no, I'm getting the hot. No, but that's awesome. I don't really make curry. You're the first one to bring that one up. I don't think I've ever made a curry. John, maybe you have. I don't love it.
Of course I have. I'm all over the place. I always like to try and make new things. I did this most amazing thing at the weekend. It's like it sort of blew me away in that. When you're making like a curry or a stew or whatever, you're building it in stages and you're building flavors and you see how it all comes together. This recipe is basically get like a casserole pot and smear it like a deep in butter on the bottom and on the sides, and then you have sliced onion, sliced beef, sliced onion, sliced beef, and basically then put a cartouche on top of it, put the lid on it, and stick it in the oven. And you think, I haven't done anything to this. and like 3 hours later, this thing comes out. All the liquid comes out of the onions, and it's on the final bit of it is it comes out. You got all the liquid out of the onions and the beef, and it's just this amazing. The beef's all tender and everything. And then it's the last piece is there's some flavoring stuff like garlic, parsley, chili. Can't remember what else. A couple of other things. You just stir it through, and it has the most amazing flavor. I was like, because like I said, I'd never done it before. I like to try new things. I'm saying to my wife, this can't be right. There must be something I have to do. What's going on? It's like alchemy, like this is amazing.
You're making me hungry, and I have made a curry dish one time in like, it doesn't count. I made it in one of those like Instapots, so that doesn't really count. But when I make a roast, it does take me all day to do, John, and I put onions in there, and it does seep in like that. But Neil, this is all about you right now. Anything else about your about the dish you like to make, or we can move on to the next question?
No, I'm happy to move on to the next question.
Well, tell us about your favorite adult beverage.
And again, it depends on the weather and the location. I like a good British real ale, just as Americans don't like it, warm and not too fizzy.
No, I like a Boddingtons. I know that's not quite what you said there, but I do like a good pub ale. Most Americans don't like that, but you like a casket. You like a room temperature beer. Good for you. All right.
Yeah, but then if the sun's shining, a nice gin and tonic or a
Oh yeah.
Campari spritz, or if the mood takes, it might be a nice glass of wine, it might be a nice glass of whiskey. It
Nice.
depends on the mood and the location.
No, we're all good. That just
So,
shows that
so I'm going to quiz you a couple of those. Gin of choice.
Gin of choice. That's a good one. My favorite is my homemade rhubarb gin.
Okay.
Rhubarb and ginger gin. So you just infuse homegrown
Sure.
rhubarb
Yep.
and ginger with some gin for a month or so with some sugar, and it's beautiful.
That sounds delicious. And then, so favorite whiskey.
Whiskey, I tend to be a Highland kind of.
Okay.
Oh,
Okay.
yeah.
So when you say Highlands, that's Speyside or
Yeah,
yeah, yeah. So I'm gonna. I know I'm very conscious of time, but I'll try and say this very quickly. My wife and I were back in Edinburgh before EBF. We went for lunch at a restaurant, and at the end of lunch, every table had everyone finished off with whiskey at the end of lunch, and so the restaurant would bring out the bottle, pour it, and then leave the bottle on the table. And so I was on antibiotics; I couldn't drink, which was incredibly frustrating. But then it was like trying to piece together what other people had, and one which I'd never had before was that someone had this little dark black squat bottle, and we figured out it was Bunnahabhain, which is an Islay whiskey, and it's not as peaty as a lot of Islay whiskies. Absolutely delicious. I was much more because I grew up near the Auchentoshan distillery. It's a lowland distillery, and then know the Speysides because we used to go and obviously have trips up there. So this was a new one on me, and that's my new go-to is the Bunnahabhain, except I have no more.
Sounds good, John.
Yeah. Anyway, let's move on. Weekend plans. Do you have any big ambitions for the year as well?
I've not thought about ambitions, other than I think it's going to be a breakthrough year for diagnostics and patient-centric sampling this year. But this weekend, we're off to see a comedian tomorrow night, Ed Burns. You probably won't know him in the US.
Sure. Yep.
Very funny guy. Saturday evening, we're going out for a Burns supper, so looking forward to that. And then in a week's time, it's my wife's birthday, and falling on her birthday, the little tiny venue we have in our town, Hartford, has got the Bay City Rollers.
My wife will be so jealous.
Well, we're going to see the Bay City Rollers on my wife's birthday. We're going to be dancing to Shangalang and everything. It's going to be hilarious.
Yeah, one of the best moments I had like that was I went to an LC conference in Stockholm. And so, like often European conferences have the conference meal and then you got the evening to drink, etc. We had the meal and then the entertainment was an ABBA tribute band. So I'm like, I'm there with my phone, like to my wife who was back in the US. And I'm listening to the ABBA tribute band in Stockholm. How fantastic is this?
That sounds lovely, Neil. That sounds absolutely awesome. Good for you. You're like a 30-year-old with that itinerary. Good for you.
No, well, I love live music, and it's so for my 60th, the big celebrations, we're going to go to this festival for tribute bands that's called Glaston Budget. So there's 3 days worth of different tribute bands, all the music you love, but a lot more affordable. So you get to see it all rather than spending hundreds of pounds on tickets.
Sounds awesome. I don't know, John. I'm good. We can wrap up.
Yeah, I'm quickly going to say I'll keep it to a minimum. The Burns Night thing, we don't really do Burns Night, but I found a recipe for doing a very alternative haggis, neeps and tatties, which is actually based. It's from a recipe book by Tom Kitchen, who's an Edinburgh-based chef.
Yeah.
And I'm doing that for the family on Saturday evening. Full bit of shortbread and and then plenty of whiskey. But yeah, let's.
Yeah, I mean, my weekend's going to be the Chiefs all weekend, all the time. So I just got to throw that in. The Chiefs are in the AFC Championship game. But John, let's wrap it up.
Just wrap it up and let Neil go off to his next appointment. So thanks very much, Neil. Yeah, that was honestly one of the most fun podcasts we've done in a while. Yeah, thanks a lot. Anything more, Dominic?
Well, very, very funny. Enjoyed having you, Neil. We'll see you in the funny pages.